Hey everyone, my name's Grant Buckner, and I'm a Bio 1802 lab instructor. Some of you guys recognize me from the Zoom meetings we've had, and others probably have never seen me before. So hey, I hope you guys are doing good. And we're going to talk about exercise 2 today, so let's get started. So exercise 2 is about microorganisms, all right? And you'll see here we have the student learning objectives. Again, these are at the beginning of every single exercise. They're in the handout. We highlight them here again, but basically this is what we want you to know and understand, and it's okay if you don't have a full grasp of that yet. And this is what we want you to be able to do by the end of the lab when the worksheet is due, or when the exercise is due, okay? So always look back over those, see if you can do those things. That's a great kind of checkpoint of do I know what's going on? So we're going to talk about the diversity in prokaryotes and protists, and there's going to be a lot we're going to cover. It may seem like it's overwhelming, but we are going to mention a lot of details that may not be that important. So just make sure you focus on the big picture. Okay, we're going to talk about differences between prokaryotes versus eukaryotes, as well as how to classify different bacteria. Keep working on your skills of observation, taking notes, describing. Continue to develop those and keep pushing throughout the semester to keep improving on those. You guys can do this. Protists and bacteria are very cool, and I hope you come away with a greater appreciation for them after this. Before we can get started talking about prokaryotes and eukaryotes, we really have to focus on microscopy, basically using a microscope. All right, this is something every year that students come in and they feel like they have a really strong foundation in it because it's a microscope, right? You plug it in, you look through it, and you twist some knobs until you get focus, but it's actually more complicated than that, and it's something that we see a lot of students struggle with. So we are going to take some time to go over proper technique. There is a virtual lab on using microscopes, so make sure you look into that. And if you have any questions, let me or one of your instructors know, okay? The first thing we're going to talk about with microscopes is the field of view. Basically, what you see when you look through a microscope, all right? It's always going to be abbreviated FOV, okay? And it's always going to be a circle. So this is our field of view right here, and you'll notice we have a pointer. Okay, this is important to note because during lab practicals, we may be asking you about something at the end of the pointer. All right, this year with us being online, it's not as big of an issue, but assuming we end up in person and you guys continue your career and you're in a lab, we want to make sure you guys always look through the microscope with two eyes because only one of the eyepieces is going to have the pointer in it. Okay. The field of view is going to be different for each of our objective lens, okay? The objective lenses are the little round lenses hanging down. We'll talk about those. Typically, there's a 4, 10, and a 40x, okay? Some of them have 100 for your beginning labs, and until someone explicitly tells you to use the 100x, don't touch it, okay? We're only going to talk about the 4, 10, and the 40 for this lab. So basically, when we look under our 4x, that's our lowest level of magnification. It's our least amount of zoomed in, right? And you can see if we look here at a ruler, and we got some millimeters here, that our field of view diameter would be 1, 2, 3, 4, 5, okay? 5 millimeters, all right? And this is going to be more important in the last video when we talk about estimating cell size, but it's just something to be aware of, okay? As we step down to our next level of magnification, which is our 10x, we'll see that we are basically just seeing a subset of our former field of view, and we're magnified, okay? We're seeing less, okay? We're more zoomed in, okay? So what we have here is 1, 2, maybe a half, maybe a quarter, more likely a half. So we have 2.5 millimeters for our field of view. And then you guys could have probably have guessed that by the time we get to 40x, we're dealing with a super small space in here, okay? So we're no longer going to use a ruler to measure that because we can't. All right, and we'll talk again about that in the last video when we're talking about cell size, okay? What's important to also note is that as you increase the magnification, obviously we have a lower diameter of field. Our field of view is going to be smaller. We're going to see more detail, and the object also may appear a little darker, okay? So as we're zooming in, we're going to be seeing smaller and smaller portions, and that will also mean we're seeing less light. So it's not unusual for you maybe to have to brighten up your microscope as you go down in magnification. just keep in mind that if you're on the 40x and you got that brightness really cranked up, if you roll back to the 4x, you might blind yourself, not temporarily or anything, but you may see little spots. So just be aware of that when you are using a microscope, to just keep an eye on your light levels so you don't blink and get a little dazed. For a proper microscope technique here, so the parts we're going to talk about is the stage, okay, which is the black thing right here. We have our clips for the slides, okay, so this is where your slide goes. These are our objective lenses. We can only see 2 of them here. Our ocular lenses are up here, what you look through, and then we can't see them because of the stage, but there should be some coarse and fine focus knobs back in there, okay? So we want you to start always at the 4x, because you always want to start out the least zoomed and work your way down, okay? You also always start with a stage all the way at the bottom. So when you move that coarse focus, it's going to move this stage up and down significantly. So always start at the bottom. So you only have to twist one way, and you only have to zoom one way until you get in focus. All right. If you start in the middle, sometimes you don't know if you need to go up or down, and it's just going to cause you a lot of extra time to hunt around and get to the proper focus. All right. Your coarse focus may get you in perfect focus, or it may get you in a rough focus where it's still a little blurry. And that's the case, then we need to switch over to our fine adjustment knob, which will typically be on the outside of the coarse focus. And you could twist those around to get it in crisp and clear focus. Okay, the next thing we do is we move down to the 10x if we want to be more zoomed in. Okay, when we do that, we're never going to pull on these lenses. Okay, these are really expensive and they can break. We're going to use this rubber dial here, and it'll just simply twist into place. All right, the 10x usually has a yellow stripe. All right, our big disclaimer here is, as soon as you move away from the 4x, do not use the coarse focus knob. Okay, at that point, you're only going to touch the fine focus. All right, and if you're rolling the fine focus knob around a lot, that means that you never really had it in good focus to begin with at the 4x. Okay, it may seem easy, but when we're looking at something as tiny as bacteria and archaea, it's going to be really, really tiny and really, really small things that we're going to see at the 4x. Okay, and again, you may need to increase your illumination. All right. The same rules apply to the 10x as the 40, so we're not going to use our coarse focus knob. At this point, the lens will be close to the stage, and you could end up breaking the objective lens, and these are expensive, especially when we have to put a lot of students through the lab. Okay, not a big a deal this year, but in future courses, this is all good information you're still going to need. So never use the coarse focus. And then again, it's the same rules. If you're using the fine focus and you're really struggling to get it in crisp, clear focus, then you should probably just back up to the 10x or even the 4x and start fresh, okay? And again, always increase illumination so your image can appear nice and bright. All right, now that we've talked about microscopes, they're super important. I work with aquatic insects, and I spend probably about 90% of my time doing, when I did my master's thesis, 90% of my time was staring through a microscope, and there's lots of cool tips and tricks you'll learn as you continue with microscopes, but it's just a great foundational tool to be able to use, and it could end up getting you a job one day as well, if you're good with microscopes. Okay, now we're going to switch gears, and we're going to talk about cell theory. Okay, this probably isn't new to you. If it is, that's fine. You're going to hear more about this in the lecture too. But cells are the basic unit of life, and all living organisms are composed of cells. Okay, so pretty simple. The two cell types we're going to talk about, so when you hear cell types, you know, what cell type is this or this, you need to think prokaryotic or eukaryotic. Okay, those are the two main ones we're going to talk about. Okay, and we also want to talk about what they have in common and what's different. Okay, so what they have in common, you can see down here, we've highlighted it for you, is they're similar in structure and function. Both have a plasma membrane, both have DNA and ribosomes. All right, we have our tree of life here. We can see that our bacteria and archaea are our only prokaryotes. Okay, so we're the domain archaea and the domain bacteria, if you think back to last week's levels of biological classification. So we're way up there. And those two domains make up our prokaryotes, and sorry about that, and everything else like our fungi, animals, plants, algae, and protists are going to be eukaryotic, okay? Really basic, but we can get lost at at this level trying to decipher what's an archaea, what's bacteria when we get further along talking about some of these different groups, okay? So just keep that in mind, be able to know that bacteria and archaea are prokaryotes. When we talk about distinguishing between the two cell types, the first thing is size, okay? Prokaryotes are super, super small, okay? If you're looking through a microscope, even at 40x, they're going to be tiny, all right? And if we look through eukaryotic cells, if we look at eukaryotic cells, they're going to be larger, all right? Prokaryotic cells lack membrane-bound subcellular structures, whereas eukaryotic cells have them. And since they have them, they can do their internal chemistry in those subcellular structures. And with prokaryotes lacking them, they're going to use the cytoplasm. And they're also going to have a cell wall as well. Prokaryotes have a single circular chromosome, whereas eukaryotic cells can have multiple chromosomes subdivided. So some can be in the nucleus, like linear chromosomes. They can also have circular ones in the mitochondria and the chloroplast as well. Here's a nice little graph of cell size. So we can see here, electron microscope, that's not what we're looking at. In our lab, we're looking at light microscopes, and we can see the relative range. So our typical prokaryotes are down here, 1 to 5 micrometers, and it's about the same size as a mitochondria. And then larger up here will be eukaryotic cells, like typical eukaryotic cells. You can see pollen grains, things like that. So again, eukaryotic cells big, prokaryotic cells small. All right. And what do you think you're going to see? Like, how big will prokaryotic cells be when you look through a microscope? Maybe you've looked at some of the material for activity 1 already, maybe you haven't. All right. And then what are they going to look like with eukaryotic cells? Well, let's look at this example here. So we want to practice identifying this, and this is something you could do on your own simply by Googling prokaryote and covering up the screen to see if you can recognize it. All right. One thing to note is the objective we're at. So we're at 40x. So we're all the way zoomed in, as powerful as we could go. Right. And if you look at this tiny dot right here, all right, you could probably guess that that's a prokaryote, and you would be right. Okay. We can't determine that there's any features going on in there that are very distinctive. All right. On the other hand, if you look at these big guys now, we can't see all the subcellular things going on in here, but it's very big. We can see some green, so we might have some chloroplast going on in there. All right, and that's most likely a eukaryote. Okay. Now we're going to talk about prokaryotes specifically. All right, and again, remember, when you hear prokaryote, there's 2 domains, bacteria and archaea. Okay. Despite their really small size and simplistic structure, they're critical to our environment. So nutrient cycling, super important for bacteria, super important for that. Ooh, excuse me. And also, even for us, the bacteria that live in our gut are super important for us, and we couldn't live without them. They're some of the first cellular-based life forms. They're critical in, like I said, nutrient cycling and decomposition. They're highly successful. They're super diverse. They have short generation times, so they can mutate really quickly. And also, they've been around for forever, so they've had a lot of time to diversify as well. And like I said, they're essential for metabolic functions of eukaryotes like us. They can be single-celled or live colonially in biofilms, and I'll kind of talk about what that is. But essentially, it's some polysaccharides excreted out in a bunch of bacteria to say, hey, let's all live together for a little while. Okay. So if we look here, we have bacteria and archaea. Again, you've seen this slide a few times. You can see we have a lot of groups in the domain bacteria and not as many in the archaea. When we think about the movement of prokaryotes, all right, they are motile, which means they can basically move around the environment. Some can glide, so they're kind of aided by the production of mucus. Some can swim with a flagella that can kind of flap around and propel them forward. And then there's a lot of other ones that there can do some floating and twitching, some other characteristics of movement. But most of the time, they're moving by a flagella, which is a whip-like structure that extends from the outside of a cell. So you can see here in A, we have one flagella right here, or flagellum. Sometimes they can have one, sometimes they can have many. Okay, it just depends on which group we're talking about. And that can be a good characteristic for figuring out to classify different groups of prokaryotes. One thing to note about the flagella is that it is embedded in the plasma membrane, but it's not covered in it. So we can see if our black simple drawing here, the black lines are our plasma membrane, it doesn't cover it. All right, and we'll talk about, keep that in mind from when we talk about eukaryotes, because we're going to see some differences there. Here's a nice line drawing that you might see in a book. So again, plasma membrane here, it's embedded, not covered in it. All right, other important things is that it's made out of a single helical rod of protein, excuse me. Okay, so it's all one piece. Okay, that's basically what you need to know. And we'll see in eukaryotes, that's not the case. All right, the way these guys move is described as a hook and motor. So they essentially kind of hook in there, and then they, if you can watch me for a sec, they kind of spin around, kind of like a propeller, okay? And then repeatedly spinning around will create forward movement in whatever medium they're trying to move around in. All right, lastly, prokaryotes can also form biofilms, okay? Biofilms are pretty common in the environment. They're capable of a lot of different things. The slick stuff on rocks is a biofilm. The gross, red or sometimes kind of orangish thing growing on your shower curtain or your sink, sometimes that can be a biofilm. All right, and biofilms have been studied a lot because this is also how cholera acts, so it can be spread this way as well. So what happens is we have some single floating bacteria around. They kind of throw some things out since their environment, and they find some other bacteria and they start to aggregate. All right, this is when they're secreting those polysaccharides, they're sticking together, and when they're together they start to grow and divide, and they create a biofilm, which is just kind of a grouping of bacteria and polysaccharides. As that matures, it'll just get bigger, and then eventually some of these will get released out into the environment, and then it all repeats. Okay, so I know I threw a lot at you. This is the end of activity 1 here, so this is the end of this video. If you have any questions, let me know, but I hope it went well, and I will see you guys for the next activity.